So neurotech is technology related to neuro applications, so essentially things about the brain. And so our approach to understanding how the brain works is we have a two-prong approach. One is the in vivo side and the other is the in vitro side. So on the in vivo side, it means observing brain physiology by inserting things like probes. So we have a group at the lab that is able to create these probes that can be implanted into humans and have been implanted into humans. And a lot of that is for the applications of treating things like post-traumatic stress disorder or Parkinson's or epilepsy. The other half of us work on more in vitro things, meaning we take the human physiology outside and observe it on the outside of the body. So this is a little bit like, the best way to understand something is to take it apart. So on the in vitro side of the house, we make a lot of what we call organs on a chip. And ours, obviously, because we're a neurotech group, we're interested in the brain. And so we have the brain on a chip. And what that means is that we recreate the brain physiology on this chip that allows us to understand how the brain works. So we grow neuronal tissue, such as neurons and astrocytes, on these devices that have these electrodes on them. And so we can record how the cells talk to each other. We also have electrodes that also have the capability of sensing chemical cues, because cells also communicate chemically through the release of different things. And so we're able to look at that also with our technology piece of it. So the same technology that we use for these implantable electrodes, we also use for the in vitro side. The goal of the brain on the chip is to be able to develop a model that we can then do different testing on. So we don't have to test on animals and we don't have to guess when we test on humans. One of the big problems with pharma is when they develop these medicines, having a good way of testing them before they give them to humans, because what you don't want to do is make a mistake and discover that this medicine that worked well in an animal is going to have detrimental effects on a human. The other problem, too, is sometimes we miss out on really cool medicines that could have actually helped humans. For example, aspirin, if it had to be developed the same way that any medicine now has to be developed, it would never have made it to market, because aspirin kills a lot of the rodent models. And so somebody would have looked at that and said, oh, we can't give aspirin to humans because it killed our rodents. And so we would have missed out on a drug like aspirin that actually has even benefits for cardiovascular benefits. So that's the downside with using animals, is that you still then have to translate to human. And in some cases, you can't test things even on humans eventually. Like, for example, again, I work at a national security lab, and a lot of what we develop sometimes are countermeasures to things that could hurt people. And so with these things, you don't want to test on a human. You want to know that it's going to work when the time comes. And so it's different than a medicine where you do eventually test on a small population of humans. In the example of countermeasures, you wouldn't be testing on a small population of humans. By the time you need it, and you need to know that it's going to work effectively. So that's the benefit of these organ on a chip projects, is that it gives us a way to be able to test things for the benefit of humans without actually having to test it on humans or animals.